we are happy to open these lectures for questions. Yes, Shahid, please.
Thank you. Two really, really excellent, interesting talks. My opening question is to Rocio about the transport proteins and biliary transporters. So biliary transporters, my question is not so much whether they can have a role in diagnosis and prognosis, but whether they can have a role in pathogenesis, in carcinogenesis. So biliary transporter proteins, BSEP, MDR, MRP, they regulate bile acid flow. Bile acids are deemed to be toxic and potentially carcinogenic. And we know that BSEP, MDR, MRP, etc., vary genetically amongst human beings. Could that be the second hit? So if you've got flukes or PSE or something else, that on its own isn't enough to cause cancer. You need the second hit. Could it be that if you're heterozygous for one of these things, subtle difference in your bile acids is that second hit?
I think it's a very good question, and I think it's a very interesting point. Yes, I think there are alterations in these transporters that could be associated to increased levels in the bile acids in the tissue, and we know that the bile acids are not tumorigenic, but are co-carcinogenic. So, yeah, the presence of certain bile acids could be responsible or could contribute to the development of cancer. It's true that many of these transport proteins change to protect the liver of the accumulation. So when you have cholestasis, they try to pump out the bile acids, so they are eliminated by the kidney, for instance. But in certain cases, you don't have that, and you have alteration, and you could have a different accumulation of bile acid because they prefer certain molecules instead of others. And yes, I think it could be a good point. Yeah.
Yes, please, from the floor.
Hi, thank you. I have a question for Michela. In your system, when you add the drug to the organ on a chip, where do you add the drug? And is it a possibility that the ECM that is growing create a barrier, and so the drug concentration within the tumor cells is lower?
Thank you for your question. It's very interesting. We study the diffusion of the molecules by performing a more engineeristic analysis. We performed, it's called COMSOL simulation. We take the results obtained with the FMN analysis, so the stiffness created on the 4, and we applied our dimension of the chip with the stiffness on the 4, so when it was established the major tumor niche, to study the right concentration to seed in the endothelial channel to allow to have the right concentration within the tumor niche.
So the concentration doesn't change, basically, so it is a true effect
of
the drug.
No, because they are really small molecules, so they cross easily from the endothelial to the tumor niche.
And I also have another question related to the T-cell migration.
Yeah.
Is it a possibility that the T-cell, besides the CXCL9 or your factors, that it also they also follow the ECM? Is it something that you have investigated, like staining the collagen fibers and see the interaction between T cells and the collagen fibers, if they follow basically the
No,
ECM?
we didn't check at the moment because they are the first preliminary analysis with the T cells, but it's a really good suggestion because it's possible to follow in live imaging the migration from the endothelial channel to with our chip, and so it's a really good suggestion, and we will sure check the migration and the trafficking of the T cells.
Thank you.
Thank you.
Thank you. While we are waiting for further questions, I will have one for Rocio. Your SCBT, which is specific for cholangio, I think you showed some data about the possibility to use it to modulate the entrance of cisplatin. So would it be expandable to other molecules and compounds, or does it have specificity for the different types of compounds?
We tried first with cisplatin, and this was like the first family of compounds we tried. But we are now synthesizing, we have chemists in the group, and they are synthesizing other drugs bound to bile acids. So yes, if you have the ability to synthesize these compounds, yeah, this could be used for other drugs that could be useful, I think, for hepatocellular carcinoma, cholangiocarcinoma, and even tumors in the intestine colon, because they will be in the enterohepatic circuit, and so all the tumors located there could be for them.
Thank you. Sergi.
Hi, Michela. This was really cool. It's more of a methodological question. It's like, how many cells do you need to use in each one of these cultures, and which ratios do you use to, like, for each cell type? And it's like the other question is, how do you assess chemokine expression or secretion here? Like, what technique did you use, and how many of the chips do you need to analyze every time, like chemokines?
Okay, thank you. So the chip are really now microscale because the central channel is only 400 micrometers. More or less with 1 million of cells in a 1-to-1 ratio with the tumor cells and calves, so half and half. Mm-hmm. We see them more or less 15 chip. So we have a lot of replicates with a really small number of cells. And we, at the moment, we analyze the expression of the chemokines on real-time PCR, and we need to extract the RNA from at least 3 chip.
It's just I was wondering if you do any chemotherapy treatment and cells start dying, how you recover, like techniques like PCR, like how can you recover like enough materials for the expression of this kind of chemokine rather than ELISA, and then if there are enough material to do ELISAs or other techniques?
Yes, we collect. You can collect also the medium, and we didn't do at the moment, but we are now planning to perform also the Luminex profiling that allowed us also not only to visualize the main molecules, IL-10 and so on, but also to screen several chemokines from the conditioned medium of the chip that you can collect, and we collected for each patient at a different time point of the drug treatment, also this conditioned medium.
Very last question, sorry. It's like these caps, how senescent they become while they are in culture. How do you find any evidence of senescence?
No, we didn't check, but it's a good suggestion.
Just to check, like if chemokine changes are due to, because these are patient-derived caps, isn't it?
Yeah.
And then, like, just depending on the conditions, they can become senescent rather than that.
Yes, it's a really good suggestion. We will check for sure. Yeah. Thank you.
And it was more of for Rocío, it's more of a common, maybe a question. It's actually, it was very, very interesting, and I was wondering if you have any plans to, because in ideal world, what you would do is like look at for each drug that we think that is not working in cholangio, it's like to check which is the right transporter for this drug, and then you can screen every single, ideally, patient and see like what are the levels of these transporters, and then, because what you're proving is like the drug is effective. It's not a matter of the drug is not working, it's just that the drug doesn't get into the cells, and it's not it cannot exert its function. So at the end of the day, it's like we have a lot of drugs that could be actually reactivated to become efficient in cholangio just by conjugating them to, I don't know, an antibody or just to a peptide that gets them into the cell. So do you have any plans in screening, like in looking at this coupling of receptor and drug or anything like that?
What we have is like different cell lines expressing different transporters, and with these cells we can investigate a panel of compounds, and we can do many at the same time to see this transport protein participates in the uptake of this one, and is more efficient for these transport proteins, for these drugs. So you can characterize for each drug what are the transport proteins that are good. I cannot say that a tumor or a patient is going to respond to a certain drug, but if you have the contrary, if you have certain characteristics that are not present, you can say that the ability to respond is reduced. It's like you have a patient without a IDH1 mutation, you think this is not going to respond to ivosidenib. But if you have ivosidenib, the mutation, not all the patients are going to respond. So you can predict like the lack of response if you know more about
exactly.
this.
Yeah.
So this is the next things we want to do for many drugs.
Cool.
Thank you.
Michael, I have one question about your T-cell trafficking, which is quite interesting. Have you looked, or do you think that through your chip is feasible to look at the functional changes of those T cells? So rather than just checking the recruitment, do they feel the CAF to change the way they work?
Thank you for your question. Yes, so we are now planning to perform also the recovery of the T cells after. Yes, and we want to perform the FACS analysis to analyze not only the release and the trafficking, but also the functional activity, if they become more exhausted rather than more activated, and also correlate these data according to the patient's characteristics. Yes.
Yes.
Michaela, I have a question. It's probably a really stupid question. I don't work with cells, but or chips. But so you have your chip, and when you do your drug treatment on your chip, how do you know? And you're giving Gemsys or whatever. How do you know what dose to give? How do you know what dose is comparable to human pharmacokinetics?
Oh, thank you for your question. Yes, to do this experiment, we study the concentration found in the human plasma after the infusion of gemcitabine and cisplatin. And so the doses that we tested are in the range found in the human plasma after the infusion, to be sure to test in a more reliable way the drug compound within our platform.
Okay.